Hey Bio30, so this is our first topic of our reproduction unit. We're going to begin with the male reproductive system, and then in a few videos we'll also discuss the female reproductive system. So we'll notice that the male and female reproductive system are actually, in a lot of ways initially, quite similar, and then very quickly they start to branch off into quite diverse structures, but we'll see some similarities as we go throughout the next couple of videos in the series. So really quick overview of some terms we'll talk about and some structures we'll go through here. Um, Everybody's got a bladder. This holds your urine. You've got a tube called the vas deferens. You've got another tube, which should sound familiar to you, called the urethra. So the urethra is going to take urine from the bladder and eject it from the body. You've got the penis. You've got a front couple structures called the foreskin and the glans, which is the head of the penis. You've got the rectum, seminal vesicles, ejaculatory ducts, prostate glands, Cowper's glands, all of which we'll discuss in their role in creating seminal fluid. You've got something called the epididymis, and then you have something called the testes, and I'm just going to move my webcam over there, and then you've also got something called the scrotum. So we'll kind of discuss the purpose of all of these and the roles that each of these structures plays in the male reproductive system. So the scrotum's role is to regulate the temperature of the testes. So I'm just going to move my face around all over here, but sperm are going to best form and mature and maintain their structure and function at a toasty 35 degrees Celsius. And so the scrotum itself is quite a responsive structure where it will either move away from the body if it needs to cool down to keep those sperm at a 35 degrees, or if things are cold outside, like you've jumped into a cold swimming pool, then things kind of shrink back up so they can maintain that 35 degrees Celsius. We don't want damage of those proteins in the sperm. Okay, so here's another quick view here. And I'm gonna have to keep moving my webcam around to make sure I don't get in the way. So we've talked about the ureter, which carries urine from the kidneys to the bladder. That's about a 20 topic. The bladder itself stores the urine. The seminal vesicle, the prostate gland, and the Cowper's gland are all going to play a role in what's creating your seminal fluid. And so we'll come back to these because their structure and function are very important to the male reproductive system. We've also got tubes, two of them, called the vas deferens, and they are going to extend from each testes down throughout the urethra. And the urethra is the tube that's going to carry both seminal fluid and urine out of the male penis to the exterior world. You've also got something called the epididymis, which we've already mentioned, and then, of course, the testes themselves. Okay, so let's start with the testes. There's quite a few portions of the testes, and I'll bring our attention to a couple of these. So we've got the vas deferens, that long tube. We've got the epididymis, okay, and then we've finally got the testes. Now, inside of the testes, you have a couple pieces. You've got the seminiferous tubules, and this is where sperm are made. So the seminiferous tubules are inside of the testes, and they are where sperm are actually made. Okay, so here's a cross section of one of those seminiferous tubules. I'm just going to move my webcam over here. And so in this cross section of the seminiferous tubules, you have something called a follicle, which is a diploid cell. It's got both copies of genetic information. Those follicles then undergo a process called meiosis, or reduction division, where they'll reduce from diploid to haploid cells, which will then mature into cells called sperm. And then those sperm are going to stay there until they are ejaculated. We've also got something called a Sertoli cell, and the Sertoli cell is a cell that we'll kind of chat about this in 2 seconds. I want to keep things simple right now. But so as of right now, we've got the seminiferous tubules, and this is where sperm are made. Inside of those are the follicles, which will slowly undergo meiosis and mature into haploid sperm. As well, inside of the testes, you have special kind of cells called interstitial cells, and the name is actually really good. Stitial, just think tissue, and inter means between. So in between the tissue cells. And these cells, interstitial cells, these cells that are in between the seminiferous tubule tissues, produce testosterone, and they are going to be influenced by a hormone called luteinizing hormone, LH, from the pituitary, which you might recall from the last unit. Okay, so a little bit more on our testes here. So in the testes, you've got those Sertoli cells, which are inside of the seminiferous tubules. So here's the Sertoli cells. What they do is they secrete chemicals required for the nourishment of the sperm. So if the seminiferous tubules are the site of sperm production, the Sertoli cells inside of the testes are going to produce nutrients and chemicals required for the development, maturation of those sperm cells once they've been created. Sertoli cells are also responsible for the process of spermatogenesis, which is the creation of those sperm by turning the diploid follicle cells, the two-end follicle cells, into those haploid sperm cells in the first place. Sertoli cells are interestingly, fun fact, they're actually protected from the man's immune system. And there's a feedback loop that actually tells the sperm, or the Sertoli cells rather, to stop producing sperm when the body feels it's produced quite a few. And then when those sperm are removed or die off, then the feedback loop continues and Sertoli cells are told, hey, make more sperm. Okay. So here's just a quick diagram here. So you might have seen this on a diploma. So the interstitial cells are going to be at the bottom of our seminiferous tubule. This whole thing is a seminiferous tubule, and we're looking inside of this. So there's lots of these, what you're seeing in front of you, there's lots of that inside of one testes. And keep in mind, men have two testes normally. So then we've got follicles. And then through the process of meiosis, or in this case, spermatogenesis, you're going to create haploid sperm. Sertoli cells are going to produce the nutrients and chemicals necessary to mature those sperm. The sperm themselves have a structure. So they have a head. Inside of that head, you've got something called an acrosome. And the acrosome contains an enzyme that is designed to penetrate layers surrounding the female egg or ovum. Remember, in the nucleus of the sperm, there's only 23 chromosomes. This is a haploid cell. It's only got half the information because its goal is to join with a haploid sperm to create the full 46. The mid piece has a mitochondria. And then the final third section is the tail, which uses a flagellum. So it's got a tail whip-like motion for motility. So the lifespan of a sperm inside of the epididymis is many years. Inside of semen at body temperature is 1 to 5 days. And you could store them by freezing them at minus 100 degrees for many, many years as well. That's what happens with sperm donors. So the pathway of sperm in the male reproductive system is as follows. It's going to go from the seminiferous tubules inside of the testes. It'll go throughout the testes, exit the testes, and enter that epididymis, which is this structure right here. At the end of the epididymis, they're going to enter the vas deferens, which is the long tube. By the way, the vas deferens is actually the site of a vasectomy. So it's called a vasectomy because the vas deferens is cut. So you'll cut it right about here somewhere, and then or clamp it or cut and cauterize it. And the idea is that ideally no sperm will make it past that cut. However, in a vasectomy situation, after surgery, they will say you have to wait at least like 30 ejaculations, if not more, to make sure you've removed all possible sperm from the entirety of these structures in order to ensure a success rate for the surgery. And that's, of course, no sperm exiting the male reproductive system after the surgery, after those days have elapsed. And those ejaculations have happened. So try to clear out those tubes, basically, of any remaining sperm. After the vas deferens, in the non-vasectomy situation, in the normal individual, they'll then enter something called the ejaculatory duct, which is literally a duct or an opening, which is going to create a lot of pressure through some muscular contractions to push that ejaculate or seminal fluid out through the length of the urethra inside of the penis and, of course, out of the body. And so the acronym that we could use here is STEVE-U. So seminiferous tubules, testes, epididymis, vas deferens, ejaculatory duct, and urethra. Those are the structures it'll actually pass through. Sperm will pass through. Okay. I'm going to pause the video here, and then we're going to break down some of these structures and discuss the different things like hormones and fluids that are added to the seminal fluid in addition to the sperm in the next video or two. Thanks, guys.